Hey everyone, this is Samantha Shepard, and my presentation is on the evolution of the human brain. So studying the evolution of the human brain has been very difficult. The reason being that the brains of past animals do not get preserved in fossils like bones do. So most studies that were done were used top-down knowledge of previous evolution that has already been discovered to compare and contrast skull size. We also compare humans to other primates when we look at the brain itself, because we don't have exact fossils of our ancestors' brains, and some primates are closely enough related biologically to humans that society has accepted the idea of comparing humans to primates to understand how our brains have become as complex as they are today. So through this presentation, we'll take a look at the differences in skull size and shape between current Homo sapiens and our ancestors to explain some parts of evolution, along with comparing how most primate brains developed compared to human development to understand how we have become superior in cognitive and social behaviors. There are a couple of specific characteristics that I focused on studying to explain the evolution of the brain. I looked at the inequality and asymmetry of the human brain, the fossil records of human evolution, the phenotypic distinctions from evolution of the human brain, and how epigenetics may have affected brain evolution. Understanding inequality and asymmetry of the human brain will lay the groundwork for some important understandings later on. Research has revealed that the brain is not symmetrical, and nor has it ever been. Using neuroimaging studies, we have learned that specific parts of the brain are responsible for certain parts of cognition. And although it is important to have interhemispheric communication, some functions only require using one hemisphere of the brain, and that's called functional lateralization. Studies have shown that the right hemisphere is responsible for face and spatial recognition, and the left hemisphere is exclusively responsible for speaking and language processing. Most asymmetry in adult humans is distinguished by right frontal, left occipital asymmetries. So your frontal lobe is the one in the front, and your occipital lobe is in the back. Some people have protested that idea of asymmetry in that it holds no value and that it actually aids in human cognition. But it has been proven through several studies, one of them being a study of those who suffer from developmental disorders like dyslexia, autism, and some language disorders. And they found that those who suffer from these disorders show incorrect asymmetry, or even have reversed asymmetry within the cortices. So now that we have a background on asymmetry, we can look at fossil records and see how they correspond to evolution. Many of our ancestors have left fossil records of their skulls, and through that we've been able to look back to evolutionary research and correlate the shape of the skull with potentially differences in the brain. Researchers have either found natural endocasts in the fossils or have taken man-made endocasts of the remnants of and created an accurate timeline of the fossils. A couple of key skull areas we recorded to show the progression of the skull, including the petiolus, which are asymmetrical protrusions of skull areas, and Broca's cap, which is responsible for speech and language. So if you look at this table, I'm not going to explain the whole thing, but you can kind of see up at the top, the age is 2.5 million years ago, and as you go down, you get closer to current time, going to 1.8 million years ago. And if you look at the petiolus and the Broca's cap, you can see that as we've come closer to current time, it's more how we are today with the right frontal, left occipital petiolus and the Broca's cap. And the Broca's cap is really important as it correlates to language. You know, there's not a lot of other animals out there that have language like we do and that can communicate like we do. So the presence of a Broca's cap is really important. And you can see through the evolution that as we get closer to present day, the Broca's cap becomes present and the petioluses become more pronounced. So now we're going to go over the phenotypic distinctions of brain evolution. Through transformations of human evolution, the brain experienced augmentations of certain subcortical and cortical brain areas, along with enlargements of the entire brain. And through transformations of human, oops, I just said that. With pyramidal neurons composing of 70 to 85% of the neurons within the cortex, they become the focus of many evolutionary studies. Pyramidal cells have oval or pyramid-shaped somas, or the cell bodies, with a single apical dendrite directed toward the pia-mater, which is the outside of the brain, the encasing that holds the brain. It has numerous basal dendrites sprouting from the soma, and a single axon emerging from the soma. Most distinctions of pyramidal cells and other primates include the length, number of branches, and amount of spines within the prefrontal cortex. The prefrontal cortex is responsible for cognitive thinking and decision making. During the process of evolution, the human prefrontal cortex dramatically increased in size and neural circuitry, and the neural circuitry was rewired. This likely suggests how modern humans gained the intellectual complexity that they have today. Connections between cortical and subcortical areas of the brain differ between humans and non-human primates. The amygdala, being one of those subcortical areas, is responsible for the emotional and social behaviors in most primates. Compared to other primates, humans have constructed extensive connections between the amygdala and other subcortical areas, like the prefrontal cortex and the temporal lobe. When there is activity and excitation within the amygdala itself, it sends excitatory signals to the prefrontal cortex, influencing it and causing changes in the behavior of the organism. This explains how the complexity of the circuitry of the human brain results in more superior social and emotional behaviors than other primates. So now we're going to take a look at epigenetics. Epigenetics is a highly studied area, and we have learned now more than ever that there are things that are happening in our environment that are changing our anatomical brain structures. So if you don't know what epigenetics are, they are pretty much that. The things that are happening in the environment that affect our bodies in one way or another. And I'm specifically looking at how they affect the brain. So studies have shown that there are two mechanisms by which epigenetics play a role in the evolution of the human brain. The first is experience expectant plasticity, and that's called EEP. And those equipped, well, let's just go with the two. So there's experience expectant plasticity, which is EEP, and experience dependent plasticity, which is EDP. And so EEP equips the brain circuits for intake from the environment, and it occurs due to rapid synaptogenesis and synaptic pruning that take place in the perinatal stage. This is extremely crucial during early phases of development and identifying crucial and indefinitely crucial for the development of sensory systems. The second mechanism is experience-dependent plasticity, EDP, which responds to the individual circumstances of each organism. This is much more complicated to predict than EEP, because EDP is recognized by either extended periods of synaptogenesis or the obstruction of beginning synaptic pruning. In contrast to EEP, the EDP introduces the idea that behaviors fluctuate between individuals, and that the process by retaining information will also differ between individuals. Because of this, EDP does not take place in the early developmental phase of a newborn. It can occur in later maternal times. In other primates, there is an accelerated advancement of synaptogenesis in the V1 and the PFC. So the V1 is your primary visual cortex, and your PFC is your prefrontal cortex. But in humans, the prefrontal cortex is one of the last cortical areas to have rapid synaptogenesis. This suggests that more time is spent reorganizing, modifying, and maturing that area. This pattern represents how EDP assists in maturing the human brain in the optimal sequence, which impacts the development of our cognition and our behavior as humans. A study performed on, I don't know how to say these kind of monkeys, macaques, proved some different things. So they were raised in captivity. They took a look at their V1 and their M1, and if you remember, the V1 is the primary visual cortex, and M1 is the primary motor cortex. And they discovered that different changes due to the environment effects on these regions of the cortex. So scientists identified some sections of pyramidal morphology, so the changes in pyramidal cells, are more prone to the environmental influence compared to other neural regions. So just some sections of the brain are more likely to be affected than other parts. And then humans have been found to respond to stimuli differently in the cortex compared to most other mammals. It has been discovered that deep penetration of pyramidal cells constitutes for the complexity of Broca's area. And Broca's area is the area that is responsible for speech. A study on rabbits unveiled the functional lateralization of Broca's area, and relied on several mechanisms to monitor the relationship between cell structure and neural activity. An in-depth study of several different animals proved that the cortex is a product of evolutionary history. Regardless of the way a species' brain is organized, similar cortical areas in different species will respond differently to external stimuli and will respond to epigenetic factors differently. So now let's take a look at genetics. The human genome is constantly changing. We're constantly gaining and losing genetic information. The general pattern of gene expression is very similar between humans and chimpanzees. However, specific gene expression during certain developmental formation of a fetus is different. And through time, the gap between the gene expressions in humans versus chimpanzees increased. The biggest difference now being about 10 years. This caused a gap in the amount of time spent on synaptogenesis during the early developmental stages. Human synaptogenesis lingers for about the first 5 years of an infant's life, as where with chimps, their levels of synaptogenesis decrease early in the postnatal period. So synaptogenesis, I've used that word a lot. Synaptogenesis is the creation of new neurons, or new synapses, so new connections between neurons. And so with humans having synaptogenesis for about 5 years compared to chimps, that just shows the evolution of how the brain is developed, in the sense that because we have those more synapses, we are able to make more connections, resulting in more cognition and more social behaviors. So in conclusion, the human brain is one of the most complex and thoroughly developed organs that exist on this planet. And through extensive comparative research between humans, non-primates, and other mammals, the evolution of the human brain is being more understood. The interesting studies of fossil skulls of evolutionary ancestors give rise to the idea that the changes in skull formation is an explanation of changes in the actual brain tissue. The research of pyramidal cells and their evolutionary changes between humans and other primates explain the complexity of brain intelligence and social behavior. The correlation of the environment with the effects that occur in the brain explain how evolutionary processes have changed humans to become the fittest beings on this earth. Epigenetics have played a huge role in influencing the brain alterations that have occurred through time. And so I lost my spot. So yeah, I just totally went blank. So you can see through all this that the evolution of the brain, how the evolution of the brain has come to take place. And with comparing humans to monkeys is the closest thing we were able to really see to compare evolution and to see how we've come to be who we are today. So let me know if you guys have any questions. Thanks.